Hello everyone. Today I am going to discuss about cloning vectors. A cloning vector is a small piece of DNA taken from a virus, a plasmid or the cell of a higher organism that can be stably maintained in an organism or into which a foreign DNA fragment can be inserted from cloning process. Most vectors are genetically engineered. Features of cloning vector. Cloning vector should possess an origin of replication so that it can self-replicate inside the cell. Cloning vector should have a restriction site for insertion of target DNA. Cloning vector should also possess multiple cloning sites, so it should have a selectable marker with an antibiotic resistance gene that facilitates screening of the recombinant organism. The size of this cloning vector should be less than 10 kb in size. All commonly used cloning vectors have some essential features. Origin of replication. This makes autonomous replication in the vector. ORI is a specific sequence of nucleotide from where replication starts. When foreign DNA is linked to sequence along with vector replication, foreign DNA also starts replicating within the whole. Cloning site. Cloning site is a place where the vector DNA can be digested and desired DNA can be inserted by the same restriction enzyme. Restriction enzyme recognizes the recognition site and cuts the DNA for insertion of foreign DNA. It is a point of entry or analysis for genetic engineering work. Recently, recombinant plasmids contain a multiple cloning sites, which are called as MCCs, which have many restriction sites. Selectable marker. Selectable marker is a gene that confers resistance to particular antibiotics or selective agent that would normally kill the host cell or prevent its growth. A cloning vector contains a selectable marker which confer on the host cell an ability to survive and proliferate in a selective growth medium containing the particular antibiotics. Reporter gene or marker gene. Reporter gene are used in cloning vectors to facilitate the screening of successful clones by using features of these genes that allow successful clone to be easily identified. Such features present in cloning vectors is used in blue-white selection. Here you can see the history of cloning vectors. Cloning vectors are many types. Cloning is first performed using E. coli. In E. coli, cloning vectors include plasmid, bacteriophages, cosmids, bacterial artificial chromosomes which are known as BSEs. Large DNA fragments and some organisms like yeast are not present in the cloning vectors, which are known as yeast artificial chromosomes (YACs). Phasmids are also comes under cloning vectors. Plasmid. Plasmids are mostly circular double-stranded DNA in most of the bacteria. Plasmid is the first vector which is used in gene cloning. It is found in bacteria, eukaryotes, and archaea. These transfer their genetic material to other bacteria by transformation, transduction, and conjugation. These plasmid vectors have some restriction sites and marker sites. For example, AMPR, LAG-Z are some of the plasmid vectors. Plasmid range in size from 1 kb to 250 kb. Advantages of plasmid vector. These are small and easy to handle, which are also easy for purification. Straightforward in selection and cloning of small fragments. Disadvantages of plasmid vectors. These are less useful for cloning large fragments, which are greater than 10 kb. Cosmids. These are circular double-stranded DNA. The cosmid DNA does not code for phase proteins and host cell lysis. It does not involve in multiplication of phase particles. It is packed within protein coat of bacteriophage to form inactive phase particles. Cos site is a prerequisite for packaging of cosmid in phase protein coat. It is normally used to clone large DNA fragments between 28 to 45 kb. Bacteriophage. These are the viruses that specifically infect bacteria, and during infection, the phage DNA injects into the host cell where it undergoes replication. The bacteriophages are simple in structure and consist of DNA molecule having several genes for phase replication. This bacteriophage is surrounded by a capsid which is made up of a protein. Bacteriophages generally contain lytic phase and lysogenic phase. Advantages of bacteriophage. These are generally useful for cloning large DNA fragments. And the basic disadvantages of bacteriophage is These are very hard to handle. Additional properties of vectors. It should be short, small, and these vectors should be compatible with host cell, incompatible with other vectors. These vectors should become high in copy member. It should be able to express itself utilizing the host machinery. It should be able to move under two systems such as prokaryotes and eukaryotes. Thank you.